So without further ado, I only have 15 minutes because Elizabeth knows trying to cram 15 minutes and what we're doing on the research side. The CMTA is about improving quality of life for every CMTer on the planet, and is about our incredible effort under STAR, Strategy to Accelerate Research, to find the first treatment for this disease. That's our mission. That's what we do day to day. Everyone that works at the CMTA, the board of directors led by Herb Barron, who's in the audience, the advisory board with many people here on the advisory board, and the staff, we spend 100% of our time working on this disease and trying to help everyone in this room as well as everyone worldwide. I'm going to spend the next 5 to 10 minutes giving you a snippet of where we are with STAR, or Strategy to Accelerate Research. And I hope you all walk away understanding that we've never been closer in our march for that first treatment of the disease. To remind everyone, Strategy to Accelerate Research is a process that came up off of a off-site meeting probably about 6 years ago today here in Palo Alto, changing the direction of how we looked at research and how we approach spending our capital on projects dedicated to CMT research worldwide. So, and that process includes trying to replicate our disease state in a, what I call a petri dish, or a 1524 well plate, with a tool known as an assay. And it's a tool that's designed and state-of-the-art to detect what we're trying to fix in the defective gene. Once that tool has been built, we then layer in the best technology known to mankind, known as high-throughput screening, with the ability to quickly and rapidly test any compound that man has made, any pharma company has made, anyone has made, for the desired effect. And then finally, taking any of the, what we call hits, out of that process and bringing it through the drug development pipeline. So it's a reminder that there's a method to our madness, and I'm going to go through in the next 2 slides how we have approached it and give you an idea of where we stand. So this slide that you're seeing right now, and I hope you can see it, is pretty unique to our disease, and it's pretty unique in disease-based organizations. The old way of looking at the world were funding projects, which I think of as throwing darts, that didn't have any accountability, any milestones, any metrics, and really no strategy surrounding it. And we took it and developed STAR, Strategy to Accelerate Research, by building a team second to none. And we lucked on a STAR as an acronym, but I can tell you, when you look at these 27 people that are part of our STAR network, they're the all-stars in their designated field. Now, what do we do here? We took it one step further, which is pretty amazing, because on the right side of the equation here in the boxes, you can see the disease state teams. These are experts that are within those diseases and are actually doing the work in the trenches on the projects that have been outlined by our scientific advisory board and the consortium that we traffic in. And they've all agreed on the right-hand side to be interrogated on a real-time basis by the boxes on the left-hand side. And the boxes on the left-hand side are the 5 components that we have identified are necessary for drug development. So if you think about the experts like the Shies and the Days, et cetera, that have been dedicated their life to some of the research that's going on, We now have a purpose. Our goal is a pill, a molecule, a compound, all synonyms that I want to go into our bodies to fix the defective gene that's causing the problem with CMT. So yours truly here chairs a monthly meeting where we discuss all the projects that are ongoing, and you'll hear a little bit about that from Dr. Shai in a short order, with the eye towards drug development. And I can tell you the whole concept of STAR was designed to be like a Tiffany box with a blue bow, to be delivered to a big pharma company with all the de-risking we can possibly provide them so we can get a quick answer and a quick solution. So Mike will talk about these disease states and where we stand, but I can tell you there's nothing short of breathtaking as far as the cooperation. And by the way, everyone checks their ego at the door without regard to publication. So anyone who's doing a project with a disease team, within a disease team, is talking real time. One of the things I wanted to put up here in conjunction with STAR, and we're going to make that announcement in short order as we're developing this program. It struck me early on when I jumped into this role and trying to find a treatment that the fail rate at the NIH, the big 10,000-pound gorilla, was 99%. Now, you all have your daily lives, you all have your businesses. No one can run at a fail rate of 99% for too long. And a fail rate at 99% of finding a treatment, finding a pill. And me being a layman saying, well, why on earth did we keep doing that? And you have animal laboratory models that have been treated and solved for diseases around the country, whether it's cancer, CMT, et cetera. And why doesn't that translate into human beings? So if you solve for CMT in a CMT1A rat or a mouse or a CMTX rat, why can't we solve for humans? Well, the answer is because they're not human. So we set out to try to find a way to inject what I call the human element. And with technology and the hype surrounding stem cells out there, which you read daily now, it seems, we are taking an approach in the following manner. We're setting up a program, and I would say a factory, whereby Dr. Shai at Iowa has the capability with his bank to take blood of CMT patients or fibroblasts. And fibroblasts is pretty non-invasive. It's a prick of your skin. And from that, technology over the last 24 months has been developed at where you can create adult stem cells from either of those two dynamics. Furthermore, technology allows us to take those cells now into a process called differentiation, meaning you can create the cell that you're interested in. So in 1A, for instance, we know the Schwann cell is what we want to get to. So we can go through from the iPS cells and create a Schwann cell through differentiation. And from there, our goal in using those stem cells is to create a secondary assay, another tool that we can then take any compound going through STAR and test in a tool that has the human element from a CMT patient. And I'm a betting guy, and I'm betting a lot on the CMTA, and quite frankly, I don't like losing at anything. I can tell you I think this is going to be an incredible step in the process of us finding that first treatment, because if in fact this is a compound goes through this assay and shows efficacy, I'm willing to bet that when it goes to a human being, we got a higher probability of it working. So the process that we're going to be announcing is pretty simple. We're going to bank fibroblasts in blood at University of Iowa under the direction of Mike Schei's bank. We're in the process, and it should be in the next week or so, that New York Stem Cell Foundation or CDI, which is a third-party provider for profit, will be doing the iPSC work, and then our SAB will decide who it is that will differentiate into the necessary cells that we're looking for and we want to attack. So we'll make that announcement, but I want everyone in this room to know that there's no stone that we leave unturned, and we're trying to do a comprehensive approach of everything we can to find that first treatment or that drug. Where are we now? And I think this slide gives you a pretty good pictorial of what our march is. And you can see CMT1A, which, by the way, was picked because of the science known, not necessarily because it was the most common type. But we know that an overproduction of PMP22, or 3 copies of that protein, is where the problem lies. So in that offsite, Dr. Schei and our SAB selected 1A in a list of projects that will lend itself, again, to that goal. I could tell you from the standpoint of where we stand with 1A, it's never been more exciting. We're attacking 1B recently. Brittany Wright, who's got a poster out there, is a fully dedicated person on the CMTA's payroll, basically, through the NCATS, or the translational side of NIH, is leading that effort with Mike Schei and another board member, Larry Rabbids. CMT2A, in axonal form, I could tell you we're going to be not only publishing, but announcing something that's as exciting as what the CMTA has ever done. Not to steal Mike's thunder, but for the first time, we're going to be announcing that there is an in vivo model, a laboratory model that has the gene and the phenotype for 2A. It's never been done before after millions of dollars and a lot of tries on mouse models. And we saw the pictures recently in the last week and a half, and they're nothing short of breathtaking. That's going to be a breakthrough for 2A, as well as, obviously, development of an assay to test compounds. 2E, we're in the process of taking that to new levels also, and securing the 2E mouse model that Dr. Liam has created at Columbia University, and bringing it into Psychogenics, who's doing all of our animal modeling as well as our breeding programs. I might take 1 second to let you all know, one of the major clogs in the funnel to drug discovery has been access to animal models. The CMTA took a pretty aggressive stance in saying, look, we got to own them. We can't be held hostage, or we can't have a response time that's 2 years in trying to find a compound that comes through STAR. So we've worked pretty hard in licensing the CMT1A animal models. We own rights, but most importantly, we own rights to develop anything we do commercially. It's not research for research's sake. That's not what we do at the CMTA. I think when you take a look at where we are on this, it's a major component of what we're doing here. 2 others that I can tell you we're attacking is CMTX. And I'm in the process of documenting a project we'll be doing with Dr. Martini in Germany, with another drug company in Germany that has a molecule we want to test in the CMTX animal that attacks the inflammation and the massive inflammation due to CMTX. So we'll make that announcement in short order. And then in the planning stages, we have no work that is shown on the screen for type 4, rare types, recessive types, and we'll be announcing, hopefully in very short order, a program and put type 4 on the board to attack not only 4B1, 4C, 4J, et cetera. All told, you're looking at a CMTA organization heck-bent on finding a treatment. I'm agnostic at who wins, what gets there first, but I can tell you everything's contractually obligated back to the CMTA. We have a seat at the table. Any intellectual property, any economic benefit, we have a seat at the table, because as you all know, this isn't free. And from my standpoint, if we can find a way to repatriate that money so we can fix every defective gene in CMT, that's what we're going to do. So where are we now? I put this screen up here. Last Monday we announced, and I told people last night, a partnership with Sanofi Genzyme. And what are they doing? Well, we went after months and months and months of saying, look, this is our process, here's our team, here's our assets, our assays. We are ready for you to jump on board and help us try to find a first treatment. They listened, and it's taken months. I announced on Monday last week that they've jumped in. We're investing $500,000, a lot of money for the CMTA. Over the next 24 months, they're investing $3 million. And they're going to test 2.2 million of their proprietary compounds. My hope and my dream is to have a half a dozen more where we're testing 10 compounds at a time for every disease state. ISIS, horrible name, great company. Great company, and they're approaching finding a distinct molecule from the standpoint of genetic therapy. They're looking at molecules that are DNA-based without going into the science, that really attach to the defective gene and stop the bad from happening. As a layman, that's as much as I'm going to go. We're chock full with our assays that went and our cells that went to the company, and hopefully over the next several months, we'll be getting those molecules so we can put them into the CMT animal models that we own at Psychogenics. Really exciting time. Pfizer, 3,000 compounds are being analyzed and worked on, annotated compounds. Amgen is interesting in the sense we're getting documentation finished there, but they have an FDA-approved drug that might have the ability to be repurposed for the desired effect in CMT1A. There's 3 other companies, without mentioning them in right now, but suffice it to say, from a pipe dream 4 years ago, this small little organization called the CMTA has been able to get the world experts in the disease states combined with the world experts of drug discovery to try to find a solution for all of us. I encourage you to reach out to us, ask questions, get involved. If there's ever a time to join and get involved here, it's now. I'm humbled and I'm honored to be part of such a great organization, and I thank you for your time.